Okay, so just give me a second here to get sharing the right thing and I will be ready to go. Now, can you see the right one or do I need to flip?
Just flip. We're seeing your
Yeah.
the PowerPoint.
How are we
itself.
doing now?
No, still seeing the whole thing.
Oh, okay. That's bizarre. Oh, I know what it did. Sorry. Uh, and give me one more second. How's that?
Perfect. Good to go.
Okay. All right. So, as Jason said, the DDM in the initials here stands for veterinarian, but I've done a little bit of everything over the years. I'm not sure who's all online because I can't see the participants anymore, but I may have crossed paths with some of you. I've done a fair bit of work in public health over the years, and also in antimicrobial resistance in public health a few times as well. One of my best experiences was working with Joanne Dillon on a gonorrhea project with one of our joint PhD students. The other thing I'm going to preface in terms of providing some context for this presentation is I am not a microbiologist. I am somebody who's been forced to learn some of the big words here, but I'm actually, like I say, a veterinary epidemiologist by training. So I come at these from a, how am I going to use these results in the real world kind of an angle, and that's how we've set up some of our projects. So I thought I would start, because I wasn't sure who was all going to be online today, with a little bit of a review of traditional genomic approaches to antimicrobial resistance surveillance, just to put into context the data from our actual project that I'm going to present a little bit later. I do a fair bit of work with Public Health Agency of Canada, and their go-to on a lot of their AMR stuff to date has been whole genome sequencing. And that gives you a really detailed, really fine-grained lens look at the DNA within individual sequenced isolates. So we can have a really good look at what the mechanisms are of antimicrobial resistance in those individual isolates. More and more, we see people also looking at, because they're complementary, they're not the tools that I'm going to be talking about today. I'm not sure there's any individual tool that is going to do everything for everyone. So this is a toolbox. And the other sort of big piece in the toolbox is metagenomics, where we're not just looking at an individual isolate, we're looking at all of the DNA in the sample. Historically, most metagenomics has been done with amplicon sequencing, where we amplify and sequence a specific conserved region. And the reason for this is to allow us to target in on the bacteria and to avoid a lot of the noise in the sample. It's a good tool. It gives us a really good sort of high-level overview picture of the bacteria that are in the sample, but it doesn't give us really good fine-grained information. And for that matter, it probably doesn't even really allow us to speciate the bacteria particularly reliably. More recently, a lot of people have transitioned to whole genome shotgun metagenomic sequencing, basically sequencing all of the DNA content in a sample, which is going to include all of the organisms in the sample, regardless of whether they're bacteria or fungi or whatever else they might be, and the host DNA. The challenge with the whole genome shotgun approach is there is so much host DNA in many of the samples, especially the respiratory samples that I'm going to talk about here a little bit later, that it gets very, very, very hard to find the signal you're interested in, to find the bacteria you're interested in sometimes, and to find bacteria with enough replicates of that bacteria to get a good look at the antimicrobial resistance genes. Now, a lot of folks who have, sort of pioneered this approach have taken I guess, tact of using capture probes to enrich their data by basically using DNA bait probes to hybridize with and enrich for specific sequences that they're interested in. And this gives them a lot better coverage of target pathogens and the antimicrobial genes that they're interested in. So this is one part of the toolbox and sort of the big traditional part of the toolbox. Why is this a good thing in general? Well, looking at antimicrobial resistance from the perspective of culture and sensitivity is not and should not go away anytime soon. That's our sort of functional final line. What can we see in the sample? It still doesn't tell us exactly what's going on in the person or the animal, but it's the closest we can kind of get to understanding whether a particular drug bug combination is going to behave or not. So we're not trying to get rid of that. But what the genomic data does is allows us to understand the resistance mechanisms behind that. So is it acquired resistance? Is it chromosomal mutations? Is it some sort of, specific intrinsic resistance that might be different than something that's just chromosomally mediated? What exactly is driving that? It also allows us to detect new AMR mechanisms, different strains of AMR genes, novel antimicrobial resistance genes, and other things that can and will be missed by traditional phenotypic testing, right? So the other really cool part about this, and this kind of gets into the One Health part of today's conversation, is it does allow us the detailed level of information that we need to look at transmission pathways and linkages back to specific sources. So from a One Health perspective, the level of data that I can get from doing a genomic study properly can very clearly tell me whether the problem that I'm looking at is human health specific, whether it's something that's shared across the One Health continuum. Does it have an environmental source? And not just guessing because I found the same bacteria and the same phenotypic resistance, but looking very, very specifically at the DNA in these organisms and seeing whether or not I, in fact, really do have the same fingerprint or really do have the same thing. So that comes from the high resolution and discriminatory power of these tools. The other cool thing about metagenomics and certainly whole genome sequencing, I guess, as well, is it allows us not just to look at antimicrobial resistance genes. We can at the same time look at virulence factors that are traveling together in a sample or in an organism. We can look at mobile genetic elements that are basically the things like plasmids, or in our case, integrative and conjugative elements. I can never say that. We just call them ice. Anyway, basically the suitcases that sort of package up these things and make it easy for them to transport from one bacteria to the other of the same species, and often of different species, sometimes ones that aren't even as closely related as they should be to be sharing and caring with each other. And then the other thing with the genomic data, which is really cool, and we don't often get out of laboratory studies because a lot of times we don't have the saved isolates that we need to go back and look, is the capacity to go back and reanalyze our existing data. If we have a new antimicrobial resistance gene pop up and we want to go look for it, well, that's nothing. We can just very, very quickly screen our database and go, oh yeah, there it is, and be good to go. And then obviously with that, there's the capacity for data sharing and comparability across different labs, and just having an idea of what is there at a larger surveillance network level. Obviously, with any of these tools, there's going to be challenges and limitations. There is a very real need for investment in rapidly evolving equipment. Our facilities are going to need to be changing on a regular basis to keep up with what's out there, and that can be a little frustrating from trying to figure out how to pay for all this. We need trained personnel, and their skills need to be evolving over time, and we need bioinformatic capacity, and that is both people and the computational infrastructure that's needed to run these analyses and store the data. I have lost track of the amount of space that we have consumed for our little project alone on campus here. It's pretty phenomenal. There are other challenges. There's need for additional standardization. For example, there's still at least 4 or 5 antimicrobial resistance gene databases that are out there. Some are used more commonly than others, but they all do things slightly differently, and we have attempted to merge the data across these different databases and got pretty frustrated trying to do it. We've had some success, but it's not easy. Data governance, who has access to these data and how that should be policed, what metadata should be there. The current cost and scale that is actually possible for functional metagenomics or transcriptomics, that I'll just mention quickly later, where we're actually not just looking for the DNA or RNA, but what it's actually doing, is still pretty prohibitive for a lot of the stuff we want to do. And then again, just a reminder that this should always complement but not replace our phenotypic susceptibility testing. All right, you were promised things that had 4 legs instead of 2, so here they are. I'm going to talk a little bit about a study that we did looking at respiratory disease in feedlot animals and cow-calf operations. Saskatchewan has some big feedlots. Most of the big feedlots are actually in Alberta. There's some feedlots in Ontario as well. We have a lot of cow-calf herds that send a lot of cattle to Alberta, and we feed out a fair number of them ourselves, but we have a lot of cattle in this province. And respiratory disease is the #1 reason for injectable antimicrobial use in the beef industry. It is a very substantial disease threat. And you go, why are you using, antibiotics? Well, these are not the common cold that I'm sure any of you that have kids have probably seen a snotty nose because there's been a whole week of school already, right? What we're doing with feedlots is very much like sending our kids back to school. We send them into the feedlot when they're about the same developmental age, I guess, as a lot of our kids. And they share and care, and they get bugs. The difference is the viruses that these cattle get can be fatal on their own, but very, very typically lead to substantial bacterial infections that have very high morbidity and mortality rates and have very significant impacts on the health and welfare of these animals. So this is a big deal if we don't manage this properly. We can and we do manage this properly, but we have to pay attention to the details here. So we want tools in our toolbox, and laboratory testing is one of those tools that will help us make sure that when we are treating respiratory disease in these animals, that we are using the right drug in the right animals at the right time. And there's been a lot of very large quantitative studies done, a lot of very large effectiveness studies done. We actually do a pretty good job of this. We can manage this problem pretty effectively with antimicrobials. We certainly use vaccines, but there are a number of reasons why vaccines don't completely address the issue, at least not at this point in time. Know that there's no sort of really quick fix there, so we need to continue to pay attention to make sure that we're using antimicrobials the very best way possible in these animals, and especially in these feedlot cattle that are at very high risk. Obviously, as with all antimicrobial use in human and animal medicine, there is a lot of international scrutiny and a lot of attention being paid. Back in 2018, the WHO put together some guidelines on the use of medically important antimicrobials in food animals. and basically said that antimicrobials that they designated as being critically important for human medicine shouldn't be used, with the exception of when bacterial culture and sensitivity said that there was no other treatment option. McDonald's, which has been one of the true leaders, I think, in antibiotic stewardship and actually responsible animal health and welfare in the industry, has developed some guidelines around this as well. The problem is culture and sensitivity takes 5 to 7 days, especially when we're shipping samples from the field. It's not like we go to the lab and we grab a sample and that gets analyzed the same day. That sample gets collected and then it needs to get shipped to the lab. So there's at least 24 hours, usually 48 hours transport time to get it to the lab. And then the lab has to do its thing and get the results back to the vets. So and by the time you get antimicrobial sensitivity or susceptibility testing results, it's 5 days most of the time, and sometimes it's a little bit longer if you're on the wrong side of a weekend. So this is not a per animal, individual animal quick fix to this problem. Just to update a little bit more on the current international landscape here in terms of what drugs we're actually using in beef production. The ones that we are using the most often here, and I don't know if you probably can't see my pointer, but I'm going to try anyway, are in the orange column here. The amphenicals and the tetracyclines are the ones that get used the most often. There's penicillins are used not so much for BRD, but for some other diseases. Macrolides are used a lot as well for both control and treatment of respiratory disease. And then there are some there's one third generation cephalosporin, and there's 2 different fluoroquinolones that are used as backup treatments in these animals. They're not first-line treatments. They're treatments that are used typically in the case of treatment failure. So what are we doing? Well, as I sort of hinted at, labs are currently using culture for the target bacteria and resistance testing for both surveillance and to inform treatment decisions when veterinarians are asking for this. There's also qPCR for some of the harder to culture bacteria and most of the viruses that we're interested in trying to test for. The problem with all of this is most traditional testing is very targeted. Basically, if you're not looking for the bug, you're not going to find it. With the small exception of, with culture, there's certainly some latitude there, but for the most part, our list of common tests is relatively short. But what we're truly finding in these animals is a lot bigger and a lot more complicated. So this particular cartoon was shamelessly stolen from Anatoly Trojimchuk, a good colleague of mine, and I really like it because I really think it tells a story here. With conventional testing, we've got a fishing line in the pond trying to figure out what's going on. With metagenomic sequencing, we can use a net and do a much better job of describing the population of organisms that we're after. It does characterize all of the DNA in the sample, and it can help us identify both the known things that are on our list that we target routinely, but also previously unreported pathogens or ones where we just don't usually test for them. We can also go back, as I mentioned before, and reanalyze these data and see what's going on. The basic steps in the process are obviously our sample collection. I'm going to talk about, we've introduced a step after this that I'll mention in a little bit here. You extract your genomic material, usually that's just DNA, but sometimes when we're looking for viruses, we're going to extract DNA and RNA, all of the genomic material. We're going to do host depletion. We're going to do cDNA synthesis if we're trying to find RNA viruses in there. We're going to do some size selection. We're going to do our library prep and our sequencing, and then our bioinformatic analysis. Again, with the bioinformatic analysis, There's a bunch of different steps and possibilities here, but mostly we're just looking to find what bacteria and viruses we can actually find in these samples, what antimicrobial resistance genes, mobile genetic elements we can find. Those are the things like the plasmids and the transposons and the ICE elements that I mentioned earlier, virulence genes, and markers that might help us get a better idea of some of the specific types of species in here, not just the species level, but specific serotypes or other well classified types of some of these organisms. So the project that we undertook was intended originally to develop and to refine practical testing strategies that we could do at scale, large scale, not one sample at a time, because these feedlots, we need to be looking at population level disease management, and to have a diagnostic support network that would actually allow us to integrate that in the long term for the livestock industry. So we set up a system where we could collect samples, we could get these to the lab, we could sequence them. We've Matt Links and his folks have set up a system where we can manage and we can analyze these data in a very efficient manner, especially compared to where we started. We can do that very effectively at large scale. We can get information that can be taken back to inform pen level management. The veterinarians can use that and use that to inform treatment decisions. And we've actually been able to complete the loop and show that it does actually inform meaningful treatment decisions in these animals. So what sort of considerations did we need to take into play when we were trying to put this process together, trying to find something that could be done at large scale, that could be done cost effectively, and could be done reasonably quickly? So the first question was sort of whether we wanted a process that was culture independent or not. I'm going to talk more about that in just a second. One of the big challenges I mentioned when we're doing metagenomic sequencing is dealing with the issue of host DNA. How are we going to be able to find our needle in the haystack, right? That's when the host DNA is the haystack, and mostly what we're looking for are sort of very small numbers of needles. Sometimes they can be large number of needles, but they're hiding in a lot of host DNA. Short read versus long read sequencing, which one's going to work better for what it is we're doing, and then, or hybrid sequencing where we use some data from both. And then our bioinformatic analysis, are we going to look at the raw reads, or are we going to look at trying to assemble those reads? So what we ended up doing was actually using a combination of culture-based enrichment and metagenomic sequencing, long read sequencing, filtering out our off-target host DNA. We were using Anatoly in particular, and Matt Links are very interested in adaptive sampling to improve that even further, and have had some good success in that space. And then most of our analysis to date has been done on the raw reads, and we've got some really, really cool stuff there. But we have also done some work with assembling those reads to sort of get even a bigger picture story. Now, this non-specific enrichment, what's the story with that? Well, when we first started doing this, these are healthy animals, and we were running into a situation where we had up to 90% plus host DNA, sometimes even more. So how do we give the bacteria a chance to be able to get a good look at them? Well, our laboratory manager had the idea of Stacy Lacoste had the idea of, let's just try some non-selective bacterial enrichment. Let's give them a few hours in the incubator and see what they do. Well, oh my goodness, did that end up working well. It substantially increases the proportion of bacterial DNA that we needed for sequencing relative to the host. We're seeing the bacteria that we expect to see in roughly the ratios that we expect to see. We're seeing increased detection of our BRD pathogens compared to unenriched samples, and we have substantially enhanced detection of antimicrobial resistance genes within individual speciated reads of our bacteria of interest without any need for assembly. So we've got reads, and I'm going to show you some later, that are actually long enough that we have multiple antimicrobial resistance genes on a single speciated read, which is incredibly informative in terms of antimicrobial stewardship. And this has nothing to do with the fact that it's coming from a cow. It's a method issue. It gives us a really good balancing of processing time, input costs. We can run these for just a little over $100 a sample and get absolutely beautiful data off of them. And we can generate the on-target data that we need to do the job. So this has already been published and is out there. Some of the other stuff that I'm going to talk to you about in a minute is just basically going out the door for publication now, so I'm not going to get into a lot of details. For those who are not familiar with it, we're using the long read sequencing that I'm going to talk about the most here is Oxford Nanopore, where basically you've got a motor protein that attaches to the DNA and kind of spits it through a pore in a membrane, and that changes the current in the membrane, and that allows us to figure out what base pairs are there. The current iterations of these, when this first came out, there was a fairly significant difference in the accuracy compared to short read or Illumina. That difference is a lot smaller now. And I would argue is probably not that substantial at all, given the length of sequences that we're dealing with and the weight of evidence that we've got. So we're getting some pretty good data off of doing this. For those of you who sort of will insist on being concerned about that, we have compared this to another long read platform that's available on campus, PacBio, and are getting basically identical results. Very, very, very similar results from the PacBio, which does have an accuracy. It's a different platform, more expensive, but does have an accuracy that rivals, in fact, potentially even exceeds Illumina in some cases. So what did we do here? Well, we figured out a way to extract the DNA to preserve the fragment length. We figured out a way to deplete the host so we could see what was going on. Now we're up to about 90% bacterial DNA instead of 90% host in a lot of our samples. We use DNA size selection to optimize the samples that we're looking at. We've got a library prep that is scaled and was automated in terms of being able to reduce the cost. There's a large flow cell that we can use so that we can barcode and sequence up to 40 samples at a time. When we've done whole genome sequencing, we've taken that to 60 or 80 samples at a time. When we're doing metagenomic sequencing, we keep the numbers a little bit lower. All of that together is sort of what's giving us the good data at about $100 a samples, or a little bit more than that now. And then we've got our bioinformatics protocol that allows us to run these analysis in a few hours. When we started, it took us days to kind of plod through the data that resulted from this process. The other thing that we've done that I think is kind of cool for any of you who are more epi analysis geeks, we've used Bayesian latent class models that allow us to evaluate the sensitivity and specificity of diagnostic tests in the absence of gold standard. So you need data from multiple populations with different prevalences. You need a couple of different diagnostic tests, but it basically allows you to get a really good assessment of the sensitivity and specificity of a protocol without having to assume that one of them is correct. And so we have compared metagenomics to culture, to antimicrobial susceptibility testing, to qPCR, and in fact, even to another test, recombinase polymerase amplification. And we have done some comparisons to whole genome sequencing. So we've got a bit of everything. And then I also mentioned we've done the PacBio studies as well. So trying to get moving here just to wrap things up. Bottom line is no test is perfect, but we've got very similar performance to culture for identifying the bacteria. Traditional testing identifies a few more samples with resistance and sequencing, at least in most cases, but metagenomics identifies many more types of bacteria and resistances within a single test than we get from traditional AST when we're doing MIC plates on these things. So we've got a lot A little bit less depth, but a lot broader picture of what's going on. There's a few other cool things that we've got out of what we've done so far. Calves' noses are very interesting places. One, in terms of where they're walking, and they don't really have a hole. They're not really shy about where they stick their noses, so we see a lot of interesting things. But we see the bugs we typically culture very nicely. We see subtypes of those bugs, the ones that are more likely to cause disease. We can get information down to that level. We see many types of bugs that are harder to culture and that don't respond well to the enrichment phase because they just don't grow fast enough. For example, Mycoplasmopsis bovis and Mycoplasmopsis disbar. And I know there's a lot of mycoplasma type organisms that are important in human health. We can see them very nicely doing this and get a really good look at them. We also see other bacteria that just happen to be there, bacteria that cause pink eye. There's bacterial pink eye in cattle, just like there is in people. We get a really good look at that in the AMR profile of those. We also see a lot of fecal bacteria. And I would be a little shocked if we didn't see some fecal bacteria in people noses too. But you certainly see them in cattle. Some of them are important to calf health, but there's a lot that are important for public health surveillance. So in a single swab, I can tell you about bacteria that cause pneumonia in calves. I can tell you about antimicrobial resistance in bacteria that cause pneumonia in calves. I can tell you about pink eye in calves, and I can tell you about all sorts of fecal bacteria that are important to public health. So that's kind of cool. A single sample, $100. I've spent $100 and I've got a lot of different information. The long read sequencing that we did goes even farther in terms of actually giving us information on resistance genes that are directly linked to the bacteria of interest. They're linked to other resistance genes, and I'm going to show you that in a second. And they're also linked to the mobile elements that help them spread. And this is on raw individual reads, no assembly required. We're seeing evidence of the MGEs, the resistance genes, and speciated bacteria with a single read. So here's two I think really kind of cool examples here. I'm just going to walk you through this a little bit. There's the antimicrobial resistance gene sort of start and end point. So think of a piece of a DNA as like a sentence in a textbook, right? A really, really long sentence in a textbook, right? Somebody who doesn't know how to edit their work and is really wordy. We've got our start and our end point for each resistance gene. So here we've got a list of the resistance genes we found on this individual read. This first set of rows here is one read that was 37,000 base pairs long. And I've got on this single read, raw read, I've got a EST gene that tells me about macrolides. I've got some aminoglycoside genes. We don't use aminoglycosides in cattle at all. We've got a sulfonamide gene. We've got some more macrolide genes. These guys travel together, and we've got evidence of that here. We've got this guy ends at 6096, and the next guy starts at 6152. So these guys, the MRC and MPHA are almost always friends, and they're almost always friends with SUL2, which is a sulfonamide resistant gene, and then we've got ERM42, which is another macrolide gene. Down at the bottom here, we've got one that's 89,000 base pairs long. That's got a cephalosporin gene on it, some sulfonamide genes on it, more macrolides, phenol genes. Fluorphenicol is used quite commonly in cattle for treating disease. It is considered a low priority drug, at least at this point in time. Its closest member that you might remember if you've got enough gray hair is back when I was in university or just starting university, chloramphenicol was still being used in people. That's probably the closest relative that I'm aware of anyway. There might be others. But so this is not considered a particularly important drug for people because but chloramphenicol was taken off the market because it causes all sorts of funky side effects in people. So it's been long gone for a very long time. And then MRC and MPHE again. So I really want this to set in. I'm going to talk about this just as I wrap up, but if the recommendations are to use the antibiotic that's least important to human health in animal medicine, if I use tetracycline here, or if I use phenocols down here, look what else I might be selecting for. What else is coming along in the package? You can't separate these, right? They're glued together. They're on the same read, right? They're stuck together. They're not going anyplace. So I sometimes worry that our approaches to antimicrobial management is a bit simplistic for what it needs to be. We can also look at viruses. I'm not going to get into the details on what viruses we saw because you guys won't care. There is a bovine coronavirus, which has nothing to do with COVID, not a close relative at all, but is very similar to the coronaviruses that cause common colds in people and kind of have the same effect in cattle. We see a lot of those. We can see the same bacteria on cow-calf herds, and I'm not going to spend any more time on that than that. Basically, our take-home message is that we see a lot of bacteria that can cause disease in perfectly normal calves when they get to the feedlot. I suspect we'd see the same thing if we could do nose swabs on kids. And viruses. There's very little resistance in these animals when they arrive at the feedlot. There's very little resistance in these animals when they leave the cow-calf operation, like very hard to find little resistance. It doesn't matter what tool you're using. We've used all of them now. Some of the strains that we see are extremely contagious, and this genomics approach allows us to trace that in some detail. And it was just amazing how explosive some of these bugs actually are in feedlot conditions. And it gives us a lot deeper look at these bacteria and viruses than any other single test. For antimicrobial stewardship at a bigger picture, regardless of what species we're looking at, we can get data on microbes that are really challenging to culture, which is kind of cool. So we can, in many cases, actually get data down to the level where we can look at individual mutations, chromosomal mutations that are actually causing resistance. For example, in Mycoplasmopsis species, that's more of an issue than genes. Finding ARGs within speciated bacterial sequences, a lot of metagenomics just says the ARG is there. Well, if I'm trying to prescribe for these animals or for people, I kind of want to know what bacteria it's in. I don't want to just know that the ARG is in the sample. I want to know what bacteria it came from, and this will tell me that. I can screen for a wide variety of ARGs and a wider variety of organisms than I can using any other method. And I can store that sequencing data and then reanalyze it later if there's additional information, and we're finding new genes on an annual basis, really, if not much more often than that. And then finally, there's the capacity to identify bacterial strains and serotypes and virulence genes in addition to just the ARGs. And I already talked about this. This is my personal soapbox, is the implications of being so able to clearly visualize these multiple ARGs on single reads. What does that do to our stewardship recommendations? I don't think we can walk around with blinders on and pretend that co-selection isn't an issue anymore, whether we're dealing with antimicrobial resistance in people or animals. So next steps, we've kind of worked our way through culture, amplicon sequencing, metagenomics. Looking at viruses is becoming increasingly, I think, attainable and practical. We had some pretty good luck with that, and we're really, really close to having a one test for all option that really does work to give us both antimicrobial resistance genes and viruses in a single analysis. And that's tough because RNA viruses are just a nuisance. And then there's a lot more, I guess, activity in the area of metatranscriptomics. So with that, I will be done. I think I am 5 minutes. I'm just barely on time. So considering I put this together yesterday, I think we'll call that good. And I'm certainly happy to, if anybody's got any questions. I apologize for the whirlwind tour.
No, that's great. Thank you so much, Cheryl. I'm just going to stop